Okay, in the previous short little PowerPoint video I gave you, we had talked about differences in plants and animal cells, and then we got into the structure of eukaryotic cells by talking about the nucleus, the nuclear membrane, and the nucleolus. Now we are ready to move forward into the cytoplasm, and we talked about, again, the cytoskeleton that is found in there. And we said one of the functions of the cytoskeleton was to help hold the organelles in place. So now let's talk about those organelles, or tiny organs. The tiny organs, or organelles, make up what is called the cellular machinery. Okay, and you can see a list here of all the different types of organelles there are. This picture you're picking at is actually, you should remember, an animal cell. So let's get going through the different types of organelles. First up, I want to talk about the centrioles and the endoplasmic reticulum. I had previously mentioned the centrioles when I talked about differences in plants and animal cells. So with that said, remember that centrioles are found only in animal cells, not in plant cells, and I told you that they play a very key role in cell division. You can see in the picture below, what you see are chromosomes being pulled apart, and then that cell will divide. Okay, what these things are they are pairs of microtubules. So the centrioles come from the cytoskeleton. They develop right before cell division begins. So they are a part of the cytoskeleton, and they only show up to help with cell division. Okay, and again, found only in animal cells. Okay, another structure that we have is something called the endoplasmic reticulum, usually just known as by its two initials, the ER. The endoplasmic reticulum, as you can see there from the picture below, usually it starts near the nucleus and then works its way out through the cytoplasm. Okay, and its primary purpose is to help move substances within the cell or through the cytoplasm. It's basically interconnected membranes that act like little hallways from one part of the cell to another part of the cell. Okay, so again, just kind of think of it as the hallways. This is the means or tunnels that help us get through the cytoplasm and move substances from place to place. Next, or something I should tell you, is that there are two forms of ER. This was in the video that we watched the very first day on cell structure. And basically it depends, does it have ribosomes attached to the surface or not? If it has ribosomes attached to the surface, this is called rough ER because there's little bumps on the ER. Okay, it transports materials to and from the ribosomes. It also will help modify some of the proteins from the ribosomes. Okay, that means it changes them. Okay, in many cases, these proteins will actually enter vesicles, as you saw in the previous video, and make their way to another organelle that I'll talk about later, is the Golgi apparatus. Again, if it's smooth ER, they have no ribosomes attached, but they do have some very specific jobs. Okay, one of the jobs is to help detoxify the cell. That means that the smooth ER helps remove toxins from the cell, which is very important to the cell's life. It also has enzymes that help build other molecules besides proteins, and it helps with the constructions of carbohydrates and lipids. Okay, so rough ER dealing mainly with ribosomes and proteins, smooth ER dealing mainly with carbohydrates and lipids and the detoxification of the cell. As we continue through, we have our ribosomes. Our ribosomes are the protein factories. Okay, that genetic material or genetic code we find in the nucleus gets taken out of the nucleus by a special nucleic acid called RNA. It goes to these ribosomes. The ribosomes can read that genetic code, and from that genetic code, they start making all the different types of proteins that we need. The Golgi apparatus, as mentioned earlier, okay, a very, very important structure. In plant cells, this is where the synthesis of the plant cell wall occurs. The Golgi apparatus will take in glucose molecules, if you remember, and it will change those glucose molecules into cellulose, and then that cellulose can be used to make cell wall. Okay, so that is one of the very important jobs of the Golgi apparatus. The Golgi apparatus will also, as you saw in the video, modify many of the proteins by folding them into certain structures. Okay, and once it has modified proteins, and it also helps in modifying lipids and carbohydrates, as you saw with the plant cell wall, it packages them and then ships them off to where they are needed. Okay, so vesicles will bring a lot of stuff in. You can see a pinched off vesicle there in the picture. Vesicles bring stuff in. Once it gets modified, gets put back into a vesicle where it can be shipped off where it's needed. So that is the Golgi apparatus. These are sometimes called Golgi bodies. Next, we have the lysosomes. What are the lysosomes? The lysosomes are kind of the cleanup crew. They contain digestive enzymes. Those digestive enzymes in turn help to break down old cell parts. It helps to digest invaders, little bacteria or anything gets inside a cell. They may help to engulf and break those down. And it helps in cell renewal. Okay, it's basically it aids in cell renewal. Okay, what it does is, each of these little organelles or different parts of the cell break down over time. New parts have to be made, and what happens, the lysosomes take the old worn out parts, break them down so that parts can be reused. Okay, we mentioned vesicles earlier. Vesicles are kind of the I don't know how to say it, the carts, the transport carts of the cell. Okay, they move materials from place to place in the cell. They can form from any type of membrane, whether it be the ER, membrane of the ER. It could be part of the Golgi apparatus. You can even form vesicles along the cell membrane. So again, these just help in the transport of materials, not only in the cytoplasm, but also in and out of cells. As I move on, excuse me, we're going to talk about vacuoles here. And now one of the things that we did mention earlier, there's a distinct difference between vacuoles in animal cells and plant cells. What are these? These are, again, membrane-bound storage sacs. The key part there being the storage. Okay, this is where we store material. Most of it is water and or waste that gets stored in there. These are much more common in plant cells and animal cells, and they also are much, much larger in plant cells. Generally speaking, a plant cell will have only one very large central vacuum, mostly filled with water, as I said earlier, and this helps the cell to the plant cell to maintain its shape and its turgor pressure, which makes the shape, it makes the cell firm. Okay, animal cells, not all animal cells have a vacuole. Depends. Okay, if they do, they're generally small. There might only be a very few. Okay, so again, these are for storage. As we move on, we time two more very, very, very important organelles. The first of those being the mitochondria. We've talked several times about cellular respiration here. At least I've mentioned it. We'll talk in much more detail about that. This is where glucose is taken, and with the help of some oxygen, we are able to make our primary energy molecule, ATP. Okay, ATP. We can also break down, I did mention this earlier in class, that we can use lipids as an energy source if we run out of glucose. Okay, but we produce ATP, adenosine triphosphate. Okay, and that's the energy that our body runs on. They have a double membrane. We'll talk more about this in class a little later on. And something really unique about the mitochondria is that they have their own DNA. We have the DNA that's found in the nucleus, but mitochondria actually have their own DNA. It's called mitochondrial DNA. Because of that, many biologists have surmised or believed that mitochondria actually used to be unicellular organisms that were engulfed by larger unicellular organisms. And once those larger organisms realized it was more beneficial to keep the mitochondria than it was to eat it, we ended up with one of our first organelles, and we ended up with the development of eukaryotic cells, or the beginning of the development of eukaryotic cells. Okay. Now, we mentioned chloroplasts earlier. Chloroplasts are found only in plant cells, and there are certain types of algae or protists and bacteria that may have chloroplasts, but these capture sunlight. We do not have these in animal cells. They capture sunlight. They have a special pigment called chlorophyll. And what that can do is it can take the sunlight, it can take carbon dioxide and water, and it makes food. Specifically, it makes glucose, that very important carbohydrate that we need for cellular respiration. Again, these things found only in plant cells, and that's what makes the leaves and stems and all that stuff of certain plants the green color that they appear. Okay, so I'm going to stop right here with this one, and then we will get into the cell boundaries, the cell membrane, and the cell wall.